Hey there folks, we'll make a start to this final session. What we're really hoping is that everyone would sort of come forward into the first couple of rows, but we understand that's not going to happen. But the idea is for this session, we want to do just a bit of a spiel on the viruses and pathogens, just talk about them, but then we want to hear what you guys are seeing in your hives. And if you've got questions about pathogens, then Richard will be happy to answer them. But so if we hope to make it interactive, is what we're saying. Okay, so keep that in mind. But we'll give a bit of background. So my name's John Mackay from Nature. We do pathogen and virus and AFB testing in our laboratory by DNA methods. And this is Dr. Richard Hall from MPI, together with Hayley, who is hiding somewhere. No, there she is. Runs the bee pathogen program at MPI. So I'll give a bit of a brief intro into some of the viruses and pathogens that we see in New Zealand, and Richard will put it on a bit of a New Zealand prevalence scope, if you like. So as was mentioned, I think, by Sammy or Juliana, there's a number of different viruses. I sort of got interested, I get interested in things that harbour a lot of different viruses for some weird reason. Grapevines harbour 70 different viruses that I used to work on. Bees harbour over 20. And with new methods, such as next generation sequencing, that people like Richard here are expert in, then new viruses are being found all the time. And that sort of thing is what spins Richard's wheels, is finding new viruses. So before Varroa, there had been few viral surveys done in New Zealand. Using enzyme methods or ELISA methods, there'd been a few viruses detected, but they didn't cause major issues. So like acute bee paralysis virus and Kashmir bee virus. And so this was some of the work done by the predecessor to plant and food research, or Hort research as it was at the time, and they looked at some of the viruses in New Zealand. They found chronic bee paralysis virus, black queen cell virus, and Kashmir bee virus. After Varroa, the viral landscape changed. Deformed wing virus was first detected in New Zealand in 2007. And when in about 2011 it was reported, or 2010, it was reported as a cause of major overwintering loss, that's really when I got interested in the viruses and pathogens. And I went to see these two fine gentlemen because they're also in Gisborne, as I was, and they fully supported me to look into the viruses and set up the first virus assays using DNA methods. So, you know, it was thanks to these two. I also took up beekeeping under Barry at the same time. So, you know, my wife probably not so happy with them. I don't know. You've heard quite a bit from me actually, sorry about that, about PCR and qPCR. And so quickly flicking through it, and I really will, the guy on the right there was literally on this road here. This is the road, here he is standing, hoping not to get knocked over, but he was literally driving along this road, and he was on his way to his cabin, and he literally conceived of this process called PCR, polymerase chain reaction. And it's a way of detecting minute amount of DNA. And if you think of people who get their Nobel Prize in 30, 40, 50 years, he got his in 7 after discovering PCR. So it's, you know, one of the great science inventions. And you've probably seen, if you watch CSI, you see these sort of bands on a gel type thing. But what we tend to predominantly use, and Richard as well, is what we call qPCR. And as I mentioned during the AFB, for those who are submitting Manuka samples for the MPI standards, you're well familiar with qPCR and the CQ. And so the lower the CQ, the higher the level of the target being looked for, whether it be Manuka DNA or in this case, viruses. So for this sort of thing here, as I said, this sample here done in duplicate, high level viruses, this sample here, lower level, all based on that CQ. 2014 was looking at the viral landscape as Varroa came in. And this was done at the University of Otago, and that's really all I'm going to say about it, because Richard will discuss this more in his prevalence work. So this was really the, we were doing our virus testing, but this was the first one that really looked at the landscape before the bee pathogen program. So looking at some of the viruses that we see, these are 3 closely related viruses. They're fatal when you artificially inject them into bees. They kill them very quickly. But we typically only see Kashmir bee virus now in New Zealand. We don't have the third one, Israeli-associated paralysis virus, that MPI did a survey on about a decade ago to prove that we didn't have it. Chronic bee paralysis virus. This sees bees trembling, and I'll show a video that just came recently from someone posting this. There's been a few incidences of this in New Zealand recently, and you can see a number of different symptoms from little black bees in your hive, and you'll also see bees in the grass. They're paralyzed and they're being ejected from the hive. They're either trying to fly and can't, or they're sick bees, and the other bees are going, get out, and they're ejecting the bees. So this video here shows, I hope you can see, if you just watch the bee, it just does especially on the top of the frames, it just does a characteristic tremble, and they will just move unusually on the frames. It's sort of just a trembling vibration. And that's a sign of chronic bee paralysis. Deformed wing virus. This is closely associated with Varroa. There is a thing of, well, control Varroa, you've controlled the viruses. But of course, you can kill the Varroa, but they've already injected the viruses into the bees. So this was the one that really got us looking into viruses, because it's associated especially with major overwintering losses. And in USA, they actually showed that Varroa was seen to be selecting for the more damaging strains. So there's multiple strains or forms of deformed wing virus, and Varroa was selecting for the more virulent forms. So when the subtypes, when the strain diversity decreased and you had the major form, the viral titer was much higher as well. So is it the same in New Zealand? We have had a brief look. There are multiple strains of deformed wing virus. That's as far as we got. But the thing is, it's usually present without symptoms. And one of the things I think beekeepers suffer from is the fact a lot of the viruses are based on, that they're named after visual symptoms. So deformed wing virus. You look in your hive, you go, no, their wings look fine. We don't have the virus. So for that reason, I suggest, with deformed wing virus, it should really have a different name, and that is hive killer 1. So this is the one really that is the major damaging one, and multiple strains in your colonies. So if there have been Varroa issues with a colony, even if you can't see the symptomatic wings, it's a good thing to keep in the background that you could have and are likely to have issues with deformed wing virus. And basically the colony will dwindle and collapse over winter. So in 2016, we discovered a new group of viruses in New Zealand called the Lake Sinai viruses group, of which there are eight species. We suspect there's at least 4 in New Zealand, and one of them is associated with hive dwindling. We found these viruses, we characterized that we have multiple strains, and then pretty much we did nothing with it because the test just went into our panel, but we haven't looked at that strain diversity, what might be affecting hives, etc. So each of these different lines, you can see the different shapes, it's always fun to do, called high resolution melting, shows the multiple strains or DNA sequences of the viruses. So this is what we do. We can talk about strains, we can talk about viral titers. This is the way we tend to do a report for samples that come into the lab. And Tammy, who's here in the audience as well, who's left us and moved to Sion here in Rotorua, has done a lot of these, where we sort of report them based on the levels we see across all the different samples we've seen, using a high, very high, low or not detect type thing, just to put them into an easy to consider format. So something that has very high levels of deformed wing virus, it's the sort of thing where potentially you might, like an AFB quarantine hive, you might look at that hive more closely rather than just parking it and leaving it over winter. So new viruses, B macular-like viruses, found in 2015, Moku virus 2016. Are you going to talk about more Moku, Richard? Okay. We've developed assays. We haven't found it, but it is present in wasps. And a lot of these viruses, which Richard's going to talk to, have been done by Richard and Hayley. Mostly Richard, I think. Hayley's managing it. A quick thing on Nosema. Nosema apis has been around for a long time. It's a question, is it the bee dysentery on the outside of the hive? Sort of recent evidence, and just talking with Mark Goodwin this morning, Randy Oliver last year, it's probably not the sign of it. But these Nosemas do lead to reduced lifespan, and I'll talk about the issue briefly, overwintering losses, and Nosema apis may also vector, so the virus is infecting the fungal-like agent of Nosema. So where you get Nosema, it's carrying in the black queen cell virus. So Nosema ceranae, a person asked a question in the last session, and I hope they'll ask it again, about resistance to Nosemas. It was only discovered to go back in honeybees to 2005. It had actually jumped from Apis ceranae. It's discovered in New Zealand in 2010, associated with colony collapse disorder overseas in the USA. And these were the sort of signs they saw: lower brood rearing, no honey, and hive dwindling. So this sort of paper here looked at the levels where you had colonies with just Nosema ceranae, with Nosema apis, or with both. And what they found was if you had one species, if the bees didn't have either species, they were about 27 days. If you had one or other, it was about 21 to 22 days. But if you had both, then the lifespan went down to about 15 days. And this is the sort of thing that we saw with the Coromandel, or what we dubbed Cororappa, because we found in Coromandel and Wairarapa, we saw very, very high levels of both Nosema ceranae and Nosema apis. So with that, I'll hand it over to Richard.
Tēnā koutou katoa, ko Richard Hall, tōku ingoa. My name is Richard Hall. Thank you very much, John, for that fantastic introduction to viruses in the world, in Africa and in New Zealand. Now, what I would like to do is talk about some recent work we've done over years for at the Ministry for Primary Industries on the presence of viruses and a couple other pathogens I'll refer to in New Zealand. So I think it's helpful when you've got the speaker up here just to understand a bit about my own background and how I came to this. Because I've worked on viruses for about 13 years, but bees are still relatively new to me, in that I've been working on this program for 3 years, but before that I worked in other human and veterinary viruses. So I thought, when I first started working on viruses, here's something that's really simple. They're really small, they have tiny genomes. I felt like I could actually memorize the genome of a virus, because they're only about 1,000 building blocks of DNA long. human genomes 3 billion. And I like the idea of simple problems because they're easy to solve. The longer I've spent in virology, the more I've realised it's very complicated. I think the work that has been done is trying to make it less complicated. The more we look at it, the trickier it is to give you simple answers. I quite liked what Sammy said yesterday, and he was quoting someone, I think, in his talk about complex problems rarely, in my New Zealand accent, have simple answers. So the MPI B pathogen program is funded from a research grant within MPI, or operational research program. And I just want to acknowledge my colleagues Hayley, who's sitting here, Fan as well as an entomologist from our Auckland laboratory, so I'm based in Wallaceville, and Lou Gallagher, who's no longer with MPI, but it was her vision to start this program, so Lou set up a lot of this work. As John said, there's a group from Otago who did some fantastic work in 2014, and I really, if I get the chance to meet Alison Mercer and Fanny Monday, would like to say thank you for the work they did back then. What they did was look at viruses in New Zealand as a varroa was moving down the country. So up the top, you can see in Northland, that's where it arrived year 2000, and it was slowly moving down the country. What this group did was take samples from colonies in each of these different coloured areas, and they looked at the viral profiles, like what viruses were present as varroa was arriving, if it had been there for a long time, or if it hadn't been there at all. So south of Dunedin, they went to the Chatham Islands as well. And this graph here, what it shows, and you have to be careful looking at it, but we've got varroa. You'll see a dark bar, so that's the incidence of varroa after varroa had arrived, which is really high, 90%. And then there's some viruses here: black queen cell virus, sac brood virus, deformed wing virus, chronic bee paralysis virus, Kashmir bee virus, and the two there, the paralysis viruses, as John mentioned, we don't have those. So if we look at each of the viruses, what you'll notice is there's a white bar which is before varroa arrived, and then a darker bar which is after varroa arrived. And what this group found was that almost universally the prevalence of viruses increased after varroa arrived. What they did show in that paper, and I'll get you to remember this, but I'll draw your attention to it shortly, is that some like Kashmir bee virus, they increased a lot, and then after varroa had been there for a while, they would decrease in prevalence, and deformed wing virus would take over. That's because when you inject something like Kashmir bee virus, it kills the pupa very quickly, and then the viruses kind of run out of material to replicate in. Whereas deformed wing virus, the pupa will be capped, the virus continues replication, makes it into an adult bee, and can spread around. So it might be less virulent to a bee, but it's better at spreading itself around. Deformed wing virus, and you can see that in this graph. It's up to 80% prevalence after varroa arrived. So in the study we've done, and at this point I would like to just acknowledge help that we've had from all quarters of New Zealand in terms of beekeepers and other scientists. So I would like to recognise the advice we've had from various technical people who've supported us, guidance from John Mackay at Denature, the USDA. They have a Bee Informed partnership. They helped provide guidance for this work. Dennis van Engelstorp, who you would have heard from, and Karen Renich from Sammy, and another lab in Canada called the National Bee Diagnostic Centre. So we modelled our work somewhat on the bee pathogen, the Bee Informed partnership, but we put a New Zealand spin on it, if you like. Also, and I'm going to mention this again, we really would like to thank the participant beekeepers involved in the study, and also the AP2 inspectors that we used to go out and get the samples. We didn't use them, but we're very grateful for the industry support and participation we had, so I cannot stress that enough. So this graph here doesn't show anything from our work. It's data from Assure Quality, just showing hive density throughout New Zealand. So we had a cohort study of 60 apiaries, and we tried to match the apiaries that we studied to the density that you'd see roughly on this map. So we had about 40 apiaries in the North Island and 20 apiaries in the South Island. And we visited them every 6 months in autumn. So we did that twice, because this project ran for nearly 3 years. We visited them in autumn twice, and then spring we went there 3 times. When we got to an apiary, we would have the inspectors look in their hives, do interviews on a tablet with the beekeeper, and we would take samples. And this is Hayley and I doing some sampling on Great Barrier Island, because we would get to go out ourselves, and that's very rewarding for a laboratory scientist to be able to connect with the people who you're trying to provide assistance, scientific support for what their industry is. So it's got me there. Hayley would pick a frame out of a hive, shake the bees into the hive lid, and then we would scoop up 1/4 cup of bees, put it in this little live bee box container, and that would be shipped back to the laboratory. We'd also take a 1/4 cup of bees preserved in ethanol, and we're going to test those for viruses, Varroa counts, and also Nosema's. And we would take 8 hives randomly from the apiary and pop them into this box to get an idea of what was at the apiary. So because this session's about viruses, I've left out the work that Hayley had done on Varroa and Nosema. And I should mention, when we're at the hive, we're also collecting lots of information about productivity, colony losses, brood comb replacement policies. Lots and lots of information was obtained in those interviews, as well as visual inspection done by the beekeeper. So I just want to show you what my life had been a bit like. When the bees get back to the lab, I would count them out. They would be snap frozen in a humane way using dry ice, carbon dioxide. Count them out, put them in a tube like this, put them in this wonderful machine called a Geno grinder with 4 6mm, 9mm stainless ball bearings, shake them up and down at 1700 RPM, and they turn into this orange juice. And it's such a great machine. You can see if you've got naughty kids and you put their toys in it, you can turn the toy into a paste, and it will turn meat into a powder. So it really mixes up the bees. We count 50 bees out. Test them using the PCR methods, the DNA, RNA detection methods that John very eloquently introduced there. And these are the ones we're looking at. So as I said, we count for Varroa, deformed wing virus, black queen cell, chronic bee, cashmere bee, the Nosemas, Lotmaria as well, and we also had a look at AFB. So for a long time, people have been asking us, what results have you got? What results have you got? I'm only going to give you a sneak peek of the results today, because what we've had to report is the level of effort we've gone to. Currently, sitting back in Wallaceville, Upper Hutt, we've got 132,000 bees stored that we haven't analysed, that have the potential for further analysis. So it's a very precious collection, if you like. We looked inside 2,500 hives and doing inspections. Both Hayley and I individually counted out 16,000 bees by hand for analysis. Between all the inspectors, they've spent nearly 1 full-time equivalent looking around New Zealand. Lots of Ks done, thousands of tests, and the 15 that we looked for. And if you sum this up, I'll just draw your attention to this, we've collected 31,000 data points. That's the equivalent of 180 years of apiary data that we can match up alongside the virus we've looked at and the other pathogens. So for the participants, we would give them back this information, and we're very, very cautious about preserving the anonymity or confidentiality of the participants who were involved, because there's a degree of trust, more than a degree of trust, coming into the apiary. So we wanted to give something back to them as we had a look. What you can see here is we tell them whether the target was present or absent, so did they have deformed wing virus or not. And we give them a bit of a commentary as well about what we saw. So we'd say, you know, in this particular round, it was down, the deformed wing virus was down in prevalence from previous rounds, where we'd see 97% prevalence, we'd see it down to 87%. And we'd rank them amongst the other 60 participants. On this graph you can see here, most of these people don't have Nosema cerani, but for the client's report, if you like, the participant's report, they have a red dot there showing that they had about 5,000 Nosema cerani bits of DNA per bee. And you can see where you were in the field. So I think that, and I think that was helpful for the participants to understand how they were going. So onto the juicy stuff. I only got another couple of slides here. To say these are the viral prevalences: spring, autumn, spring. And what we found was that black queen cell virus is present at nearly 100%. So most bees on the mainland have black queen cell virus. That's already known all around the world. It was seen in the study done by Alison Mercer previously. So it's not surprising. It doesn't mean it's causing harm to the bees, because many of these infections can be latent. It's only when they suffer a direct injection that there is a problem. So there may be no problem with that. We expect to see it. Deformed wing virus, we see high prevalence, but you can see there's a hint at seasonality there, because it's slightly higher in the autumns, and we wonder whether that coincides with the slightly higher levels of varroa that you might see in autumn as well. Chronic bee paralysis virus, we didn't really find it that much. Our inspectors would see it, we'd see it with the tests, but it wasn't a big problem. What's really curious and interesting, and we don't have the answer for, is why Kashmir bee virus had this big peak in the spring of 2017. Because if you look at the work from the University of Otago, it shows that after varroa arrived, deformed wing virus went up in prevalence, and the other viruses like Kashmir bee virus, after a period of time, start to wane. So something happened here that drove this up. I'm just going to mention these ones as well, because there's another cool finding that we've found. A lot of places you read about Nosema cerani is replacing Nosema apis around the world. We haven't really seen that in New Zealand. We see a lot of Nosema apis and we see a lot of Nosema cerani. Nosema cerani, from what we found, was almost exclusively present in the North Island. And we see seasonality as well. So Nosema cerani and Nosema apis is something that knock colonies around in the spring after winter, and then levels drop away in the autumn. This other microorganism, Lotmaria, we see seasonality of it in New Zealand. To the best of my knowledge, I haven't been able to find a paper that shows the seasonality of Lotmaria, and it's the exact inverse of Nosema. So what does all that mean? Because I'd like to give, we would like to be in a position to give answers to beekeepers, and as Paul had referenced earlier in our MPI talk, we're currently working on getting the analysis. We're sitting on a mountain of data. And you can imagine we've got colony loss data, productivity management, there's climate data we can bring into it, the density of hives we're interested in, and the pathogen burden, is to bring all those things together and understand how viruses, varroa, interact in New Zealand, and actually may provide results of international interest as well. So I just want to reiterate and make a special mention and acknowledgement to the inspectors, participant beekeepers, and the scientific guidance and help that we've had as well. So it's been, for me, a very rewarding piece of work to be involved in and ongoing. So thanks. I think we can talk more generally about viruses now. Yeah.
So are there any questions? Are there any questions from the from either talk?
Just one question for Richard. You just mentioned the work's ongoing. I assume the funding for it is ongoing too, so is that correct?
Yeah, that's right, Barry. Yeah, we're going to be doing work for the next 3 years. Yep.
Great, okay.
My questions, well, John himself mentioned that would be better asked here. It's to do with whether or not the honeybees possess the necessary genes to build resistance to the viruses they're up against, or if there's any evidence that it is already happening.
With things like Nosema cerani, it really only was found on Apis mellifera in 2005. So the chance for, as Sammy mentioned in the previous session, the chance for bees to evolve against it would be, fairly limited in that case. Apis mellifera, that's over 100 years, but we still, find that everywhere. So, it's not so much resistance. That said, we'll often find, in what looks like a perfectly healthy hive, I mean, you can talk about the antiviral resistance more, but in a perfectly healthy hive, what looks healthy, very strong hive, we can find low levels of viruses, and we know that the viruses are unlikely to be a problem. So the bees do a fantastic job at managing low levels of viruses. But the beauty of the qPCR method is we can tell per 10 in our case or 50 in Richard's case, we can tell the viral levels. And so we get an idea as to not only the virus present, but the levels of that virus.
You made me think, actually, so I spent all this time working on human viruses. What blows my mind about bees and the literature, and if you're thinking about resistance, is I kind of think about the virus as like a big panel of levers. When you read the literature, there's a lot of discussion about if I pull the deformed wing virus lever down, does the chronic bee paralysis virus lever go up? Oh, and that makes this acute bee paralysis virus lever go down. It's really, really complicated the more I look at it, and I've not seen that. When you look at human disease, you don't say, oh, if I get the flu, does that mean I don't get norovirus, and then do I get Campylobacter? It doesn't work like that. So I think that the extra complexity with bees as well is you've got the individual immune systems of the bees, but then you've got the social immunity because of the behavioural practices of the bees ripping larvae out. And I know there's another great study from Alison Mercer on varroa-sensitive hygiene where they found the genetics of those bees meant that they were better at pulling cashmere bee virus-affected pupa out of the cell, but they couldn't detect the deformed wing virus pupa out of the cell. So I think the answer is just, it's complicated. If you develop resistance for one virus, you're going to change the interplay of things, and you've got the health of the individual bee and then the health of the colony itself, different virulences. I think that's a really good question, but very difficult answer.
Do you think queen, now that we can determine viruses and whatnot, do you think queen breeders should be testing their queens for passing on viruses?
I mean, some of the standard practices, one of the things we haven't done is trying to make things very practical for for you guys to take back to your apiaries tomorrow or the next day, what to do with viruses. And so for things like, chronic bee paralysis virus, the video that I showed, the standard practice there is to requeen the hive if it's possible, depending on the time of year. And once, things like deformed wing virus, once the queen gets infected, then all the progeny, that's probably when you're going to see symptomatic bees, because all the progeny are essentially, the eggs are being laid infected with deformed wing virus. We haven't done too much queen testing, but the few queen samples we have tested, we've found a bit of black queen cell virus, although I've never seen a black queen cell, to be honest. Seen the virus, never seen the black queen cell. But not a whole lot of the other viruses. So perhaps, if there is too much of a viral load in those queens, they never make it to to an appropriate candidate, if you like, an appropriate queen. Anything?
Yeah, I mean, I probably neglected to mention before, most bee viruses are made from RNA, which is less stable than DNA. So they change a lot, which means they will evade the host immune system. So I think that's more related to the last question. Testing queens, I mean, obviously there's a destructive element to sampling because it's difficult to get them to give a hypopharyngeal swab, but there are some ways we could look at it. Yeah, I think it's an area for more work.
In the work that I've been doing on pesticides in Central Otago, we've noticed that what I think, and this is what I want you to confirm, is a greater incidence post-spraying and about 3 months later a build-up of black queen cell virus. I'm thinking that we're identifying it correctly, and I'm not actually working on the viruses, but it was an interesting observation. Am I correct in saying that they tend to be hairless and darker in the abdomen? And why is it, if that's the case, why do the other bees remove the hairs from the black queen virus affected bees and not other viral species? And have I got my facts correct?
So normally the hairless bees are chronic bee paralysis rather than black queen cell. But you know, I don't. If there's been a test done for the black queen cell, so then you know it could be showing that. But normally the black bees are chronic bee paralysis, a set of symptoms for that. The bees, it's not quite sure whether the virus is affecting making the bees hairless, or it's in fact the bees trying to eject them from the hive and nibbling them to make them leave the hive. So that's not known why why those bees are hairless and black, but it's normally chronic bee paralysis. And I certainly know the first case we found of chronic bee paralysis. We're talking about probably 2012, was down from Dunedin ways, where we saw it. But normally chronic bee paralysis is a symptomatic virus, and it's seen on the hives. Seen in the hives. But one of the things with that virus which can cause concern, and did cause concern with a recent case that went to under the MPI hotline, is that chronic bee paralysis virus, again the trembling video that I showed, can cause symptoms where the wings will splay into what they call a K-wing. And K-wing is also found it's not actually K-wing, but it mirrors or looks very close to symptoms found with tracheal mites. So when people start seeing these wings splay into a sort of K, then there's concern about tracheal mites. And if there's ever cases like that, I'll just take this opportunity now where you're not quite sure what's going on, there is the MPI biosecurity hotline where samples will end up, I guess, ultimately with you, and thus keeping Richard employed. That hotline is there to submit samples where you're not quite sure what's going on, because they want to know of incidents like that.
Yeah.
Sorry, long-winded answer.
No, thanks, John. It's just really good to encourage people to call the MPI 0800 number hotline. I mean, that's...
you remember?
Yeah, I can't. Hayley might remember it. Put you on the spot. We heard it as Stan.
Okay.
It's easy to look up, but I think, for any deaths of colonies or hives, MPI is going to take an interest in following that up.
Just to let you know that we didn't observe any wing trembling in these bees at all, and no K-wing either, and that's possibly why I've come to the wrong conclusion about what we've got. Can you actually have the chronic bee paralysis without the wing trembling? And if I have these bees that have been stored for 1 year at -80, can you still use them and do any kind of analysis with them, just out of interest? And is it interesting to you?
I think the -80 is a great temperature to store the bees if they had the problem. I think the avenue for follow-up for us usually is to have something called in to that hotline so that we could go through the proper process with investigators. But I just think on the notion of chronic bee paralysis virus and the symptoms of it, there are two forms. One is more where you have these black hairless bees, because the notion is the hair's been nibbled off them, so they're in the hive, and the shaky ones are outside the hive, but I think there is some crossover with the symptoms, so it's not... I've noticed when I started this work, you kind of look up, you want to see a laundry list of, if they've got these symptoms, they've got these things, but like many animals, and ourselves included, viruses are quite mysterious and cryptic sometimes, so they'll show different symptoms depending on how it was transmitted to them. So I think it's just it needs some kind of follow-up.
Yep.
Yeah, so.
Thank you.
Okay.
The usual case, which we get asked a bit, I'm not quite sure what's happening with my bees. I think it could be a pathogen or something, but, we're a commercial testing lab. There's a charge for testing. MPI is not, but you're not quite sure if you want to involve MPI. So if you're just wondering what to do, grab a sample of the bees, chuck them in put them in a Ziploc bag and chuck them in the freezer. And that's perfect, because you've got the bees, they're stable in the freezer or -80 for years, and then you can decide what to do. But if you leave it for a week before you decide what you want to do, and you go back and you find the hive's died and the bees have gone, or something else has happened, you've lost that opportunity. So if you're trying to buy yourself time to decide what you want to do testing-wise, grab the bees in a Ziploc and call the MPI hotline, but chuck samples of bees in the freezer. Barry.
Okay. Just another question for Richard. There is some evidence from overseas that feeding supplements like patties and the like causes elevation in pathogens. My question is really, do you have enough samples and data that you might make a correlation, yes or no, that we're having this effect in New Zealand?
Yeah, I like your thinking, Barry, and we think we will have enough to answer that. We've currently got a paper being drafted for publication that's sort of the basic kind of epidemiology, some of the results of which I've given us a look at here. Certainly these multivariate kind of analyses that look at the effect of supplementary feeding. Yeah, we think we might be able to answer some of those questions. That's really exciting.
Yeah, I had a question around the alternative varroa treatments, formic acid and oxalic acid. We got a talk sponsored by Ecritech from the company that was marketing the varroa treatment with formic acid, and they said that it might help eliminate pathogens in the hive. Do you have any experience or data on that?
No. So it's known that it's been published that oxalic acid can knock down the SEMA levels, so the fungal-like pathogens that cause the coromandel and Wairarapa, and in fact a lot of the North Island, hive decline. So I've seen that published for oxalic acid. I haven't seen it for formic acid, so I would wonder whether they're just going organic acids and just making that assumption. But I don't have an answer to that one.
Yeah, I don't have an answer either, but I know with the data we've got at the moment, it's our intention to look at how some of the treatment methods might affect levels of other pathogens other than Varroa. So what's exciting about the bee pathogen program is it may have the potential to address some of those questions. I think there are some studies that have looked at, if you get rid of Varroa, that can have a beneficial effect for, say, deformed wing virus, but then there's other studies show that deformed wing virus can persist after the removal of Varroa. So once again, it's a complicated answer, but I think that's something we would like to address with the data we've got at the moment.
There was one report. I mean, the thing of this, the long-held belief has been, control Varroa, no viral issue. As Richard said, it can persist, but deformed wing virus seems to be able to infect other things, and it's been found replicating inside chalkbrood fungus, so which makes it a mycovirus, a fungus infecting virus, as well as bees and Varroa. So to me, it comes back to the total hive strength and hive state. So rather than just looking for doing a viral survey of, do I have munted wings on my bees? No virus. It's looking at all of the unhealthy parts or potential unhealthy parts of a hive as to whether they might be with chalk brood potentially harbouring deformed wing virus, especially if you had a varroa issue with that colony.
Thank you.
Half moon syndrome. I know generally it's stress in the colony, but is there a viral causative agent for that?
I'm not looking at him, but I think Richard's looking at me, isn't he?
Yeah.
Yeah. Okay. I can see that caught my eye.
Question answered. Yep.
No, it's it is. I mean, there are varroa ones. I think to that half moon syndrome or parasitic mite syndrome seems to be a bit of a catch-all. We said we'd make this interactive. Is anyone? As Richard was looking at me, I'm looking at people like John Berry and Frank Lindsay to see what they say about things like PMS.
Half moon, requeen it, gone.
Half moon was we had a big exercise for half moon, thinking it was something else, and that was well before varroa, and it was basically put down to nutrition. So it was a poor spring. They didn't get fed well, so some of these bees, queens, were multiple laying and everything. And it was basically nutrition. It was before varroa.
Now I'm looking at Barry. Any comment you want to make, Barry?
Well, I only agree with those comments there. If you've got it, just requeen the hive. We call it a disorder. We don't know what it is, actually, do we?
Again, I mean, if people were to see those sorts of samples and, what is this? It's an unusual, what we call etiology. It presents unusual symptoms. They're the sort of bees that, please chuck in a freezer and find a tame scientist who'll have a look at them. But the frustrating thing can be is we look at a lot of viruses just out of our own interest, and people describe a really interesting syndrome or something, and you think that'd be great to test. Yeah, it died and a few weeks ago, so there's nothing to test. Any other questions, or do people want to start preparing for the gala dinner if they're going? Well, it looks like all the questions. No, Tammy. No.
You mentioned Lotmaria passum. Did you find any Crithidia? Richard?
We just use the generic assay, so thank you for your work too, Tammy, because I think it's thought to be that Crithidia mellificae, we're unsure of the status, so the assay can capture both targets. Yeah.
For those that are interested, Lotmaria passum was a new pathogen described in 2014, I think, and it was split away from something that had been around as an overwintering cause called Crithidia mellificae. So Tammy, who just asked that question, did quite a bit of work in our lab to work out, as Richard presented on the prevalence, Tammy did a lot of work on determining the prevalence and changes in Lotmaria, and I believe didn't detect Crithidia. But if you have an interest in these new pathogens, even though she's now working at Scion with a reduced bee interest level, feel free to ask Tammy about those pathogens. John?
Are they new pathogens or are they just newly discovered?
Exactly, it's a very good question. They're newly discovered. So what was believed to be Crithidia mellificae was actually renamed, was actually found to be what is it, Tammy? What's Crithidia? It's a trypanosome. It's a trypanosome, sorry. Mind blank. So a single-celled corkscrewing organism. Should get Tammy up here, she can tell us all about them. But what was believed to be Crithidia mellificae was actually found in 2015 to be a different organism. Genetically, they found differences, and they actually realised, no, it's a different organism, so we're going to call it Lotmaria passum. But then when they went back and looked at samples from the mid-2000s, they found that the organism Crithidia mellifera, that's been published as Crithidia, that was causing overwintering losses in Belgium, was actually this Lotmaria passum. So when we had the Cororappa issue in Coromandel and Wairarapa with rapid hive dwindling, we developed a new test to look for all trypanosomes because we didn't know whether we had Crithidia or Lotmaria, as it had just come out named a few months beforehand. So it picks up both but doesn't discriminate, but Tammy used a number of assays to detect the wide prevalence of Lotmaria and the absence, wasn't it, of Crithidia.
You mentioned a few of these viruses can persist in the hive once varroa has been knocked back. How do you think they're being transmitted within the colony without the main vector being there?
I think that's fairly well established for some things. So, say, deformed wing viruses can be transmitted through feces, or the chronic bee paralysis virus is just direct contact. So that it's a good question, and I think those things are quite well described in the literature. And for each of them, there's a different method, like feeding to the larvae. So that's how they're persisting after.
And I don't think we know, at what stage they might get fed to the queen, in which case, then the hive potentially goes into a very fast spiral downwards due to, all the progeny being infected rather than horizontal transfer. No other questions? Anyone want to share experiences they've seen of viruses, or do we call it a wrap?
I saw a massive amount of sac brood this year in 3 hives. I killed them, but, sent a sample to Richard. But it's just something unusual. Also, we're seeing, for years, we bred out chronic brood virus. We used to have it in the 80s, a lot of it. I used to anyway in Wellington. And we just bred it out over a period of 8 years. And we're now seeing it again. So we're seeing some of these things come back after years of varroa or something. So whether that's the genes or inbreeding of our queens or something like that. And it's a spread, where they pull the hairs off, and then the bees bleed, of course, and then the other ones are touching them and pick it up, and that's how it's spread. And so, get them out of your hives. But generally, replacing the queen works. But we're seeing a lot more. I'm seeing a lot more of it, not in mine, but other people's hives.
Okay, thanks everyone for attending, and I hope it was a good conference. And I hope people will provide some feedback to the organisers on things like the science session and the talks, so that it can be even better next year. Cool. Thanks very much.